All right, so I understand the format of the quiz was a little goofy. That's just I can only do certain things with that. So we're gonna go over it though. Okay, so we're gonna take time to go over it, and then we'll spend the rest of the time today focusing on only a few parts. And the reason why I don't want to recover or rehash all of this is because some of it really doesn't become. The importance of it isn't apparent until later on in the semester. And so we'll come back to that later on. Right now I just want to make sure that we've got, in general, terminology, and then we're going to cover a couple of other basic concepts that I think are fairly important for us to take care of now. So here's what we're going to do. I'm going to give you just probably like 3 or 4 minutes real quick. You can work alone, work with somebody next to you, yada, yada, yada. What I did was I basically took the quiz questions and I put them into a table form here. And I actually had hard copies. I left them at my office though. So if you want one, come pick one up later. Otherwise, this is actually posted on Blackboard. So it's microbial structure and function, lesson 1. It's all the way at the bottom of the list. It's just a Word document that you can, even if you have your computer out now, you can download. So go through, try your best to fill in the blanks, and then we'll take a few minutes to cover it. All right? Go ahead. Flagella, used for movement. Do eukaryotes have flagella? Some people said no, some people said yes. I would say yes, but. And here's where the caveat is. There's a difference in the structure, and there's also a difference in the how that flagella actually moves. So yeah, prokaryotes and eukaryotes have flagella, but there is a structural difference, which I'm really not going to focus on. And the only other difference is that prokaryotic flagella, they kind of move in a clockwise and counterclockwise direction, so they rotate, whereas eukaryotic ones beat instead. So they're kind of like think of it like an oar, whereas the prokaryotic ones just go like a propeller. But that's a really minor difference. All right, what is a plasmid? Said. It's not the inner part of the cell membrane. It has to do with DNA. What was that?
Short fragments of DNA.
Short fragments of DNA containing what? Genes, non-essential. And what we mean by that is, I use this analogy every year. But I'm going to preface it with I'm not a car person. I drive a Hyundai Accent, okay? So I'm not a car person. The body of the car, the engine, everything, those are necessary parts for the car to run. You need the tires, etc. But then you start to add accessories. Maybe you're going to add a spoiler. Maybe you're going to add chrome rims or something like that. Maybe you're going to get racing wheels, okay? Those are accessories, right? Those are your plasmids. They are to soup up the bacterial cell. So the genes that that are on plasmids, and every plasmid is different, but on plasmids we'll typically find resistance genes, ones that are required for resistance against antibiotics. We might find genes that provide pathogenicity to the microorganism. So that's typically what we'll find. Some plasmids, somebody had pointed out, some plasmids can be transferred between cells. That's actually true. We'll talk about that later on once we get to molecular. That's through a process known as horizontal gene transfer, and that's something to cover later. Do eukaryotes have plasmids? The answer is yes, but most of the ones that do are single-celled. So like we can artificially put plasmids into yeast, for example, and express them. Yeast do have some plasmids on occasion, but we usually think of that primarily to bacteria. Okay. C, ribosome, obviously involved in making protein, found in eukaryotes. Absolutely. There's a slight difference in structure, which becomes really important when we talk about antibiotics. But yes, found in eukaryotes. Cytoplasm found in eukaryotes. Of course. All right. How about E? What is E here? Fimbriae. And so I've already had a couple folks ask about, wait, what's the difference between Fimbriae and pili? I usually think of Fimbriae as being shorter. They are also involved in attaching to either eukaryotic cells to solid surfaces, etc. Pili, on the other hand, are more kind of like a straw in some way. They're hollow tubes. And a pili is used to transfer plasmids from one cell to another. So this, again, is part of horizontal gene transfer. We'll cover that much later on, but that is the big distinction between the two of them. And, okay. F. Nuclei or nucleoid? Okay, there we go. Nucleoid. Location of the chromosome. Found in eukaryotes? No. They have nuclei. They're lucky them. Okay. G, cell membrane. Found in eukaryotes. Of course, every single cell has a cell membrane. What we're going to move into starting on Friday is bringing some contrast between the different types of bacteria. So what I want you to look at so some of the classical experiments in microbiology actually have nothing to do necessarily with microbiology, but have more to do with molecular biology. But there is a classical experiment from the 1940s and 1950s in which a researcher had isolated two different forms of Streptococcus pneumoniae. One that was just termed R, and the other that was termed S. And the R and S referred to rough and smooth. Basically, that's what the colonies look like when you grow them up on an agar plate. The smooth, they had the capability to kill mice. The rough ones, on the other hand, couldn't kill mice. You would inject it in a mouse, and the mouse would come out perfectly fine. The only difference between the two of them that we come to find out is presence or absence of a capsule. So the smooth had a capsule that kind of had this like milky-like look to it on the top, shiny, and then the rough, they don't have that same shiny. They're kind of more of a matte finish. Here's some data from a recent, probably in the past 10 years, paper in which I really don't want you to focus on most of this. I really want to only focus on one piece. So the lab that was doing this project was looking at two different strains of Streptococcus pneumoniae. They called D39 and Tiger 4. And they had generated in each of them a mutation so that you could have an encapsulated form, which was the wild type, and a non-encapsulated form, which was the mutant. And these data here represent how frequently did they find these streptococci associated with neutrophils. And what do neutrophils do? This might be way ahead, but everybody know what a neutrophil is or what it does? It's a phagocyte. It's a white blood cell that phagocytizes microbes. So it's a second line of defense. So take a look at this data real quick. And what's the general trend between the unencapsulated and the encapsulated strains? How do those percentages differ? Or do they differ?
Ask the question, please.
Yes, ma'am. All right. So take a look just at D39. We'll take out Tiger 4 right now. D39, compare the percent of encapsulated encapsulated streptococci that are found associated with the neutrophils, and compare that percentage to those that are not encapsulated and are associated with the neutrophils. Which one more frequently is associated with neutrophils? Unencapsulated. In other words, the ones that can't make a glycocalyx. The higher the rate of association, the more likely it is that this unencapsulated microbe will be phagocytized. And that's exactly, at least one of the reasons why, in these classical experiments, they found that what they classified as rough or those unencapsulated streptococci were unable to kill mice. They were killed off by the immune system. Whereas encapsulated ones, they have a lower frequency of interacting with those neutrophils, therefore they can survive, replicate, cause disease. Mouse is dead. And then we see the same trend here with the Tiger 4 strains as well. So about twofold difference, twice as likely to interact with those neutrophils if they're unencapsulated. So this is just hitting home the point, or this is proof of principle that one of the roles of that glycocalyx is to prevent phagocytosis, to prevent detection by the immune system. Not all microbes have these, but the ones that we are most concerned with that do are typically pathogens, and there are some exceptions to that. All right, so where did I want to go now? All right, let's skip around for a minute. I want to go to some pictures. There's a lot of noises today. Here are 4 different bacteria. What I'm going to have you do for about, say 4 minutes. For each of these, I want you to write down what's the shape of the microbe itself, so what's its morphology, and also how would you describe that flagellar structure? Okay, so is it atrichous, monotrichous, locotrichous, amphitrichous, peritrichous, trichus, etc. So take about 4 minutes, and feel free to talk it out. Ah, good. Vibrio. Anybody say bacillus for that top left one? Okay, good. There's only a slight difference. What's the difference between Vibrio and bacillus? Yeah, it's got that little bean shape there, okay? So that little bend is indicative of Vibrio. This one is Vibrio cholerae, which causes cholera. But usually Vibrio, the genus that, what am I trying to say? There aren't many microbes that have this Vibrio shape. The most common ones are all part of the genus Vibrio. So it includes Vibrio cholerae, Parahemolyticus, which is often a cause of foodborne illness from shellfish, if you ever have eaten contaminated shellfish, or Vibrio fischeri. Vibrio fischeri, if you ever see a picture of a bobtail squid at night, okay? Vibrio fischeri gives off this luminescent blue color, so that it's a symbiotic relationship. It's really, really cool. Flagellar tail on this one? Monotrichous. Good. All right. Bottom left. Bacillus, good. Peritrichus. So basically hair all over, flagella all over. Okay. Salmonella enterica is that one there. So that's a false imaged electron micrograph of Salmonella enterica. Sydney.
Do you name the shape of it first and then put the...
I don't think it I wouldn't really care whichever format you do. I usually put the shape first and then the flagellar structure afterwards, but that's just the habit I've gotten into. But if on an exam you list it the other way around, I would not care.
Okay.
As far as I'm aware, there's no rules to it. Lower right. Okay, this is a good one to look at. So staphylo means what, Zach?
A lot of, at least 8, right?
I don't know the number. I think cluster, right? Cluster. These are clustered, but this one is a tetrad because they're clustered in 4s. So the difference between that and the staphylo, there wouldn't be any like regularity to the clustering. You might find some that are really big. You might find some that are a little bit smaller. This one would probably fit best under tetrad because what we see most often are these groups of 4. You could also say, I can't tell whether this is a tetrad or a sarcina, which is a ball of 8 that has this squared-like pattern under the microscope as well. So, yeah, sarcinas look like, I'll try to draw this. Yep, a cube. This is horrible. But roughly like that. Much better picture of it in the lab. Anyway. And we've got some of those. So if you want the afternoon lab section today, if you want to, we've got some of those to look at. All right, I've saved this last one because this is a debatable one. What 2 options could it be? Spirillum and spirochete. How do we tell the difference? What's that, Jane? The pointy ends is one way we can tell. Good. What else? What else can we use as indicators? Actually, in some ways, yes. I think there's a general rule of thumb there that spirochetes are a little bit thinner and have more helical turns than a spirillum does as well. This one here is a spirochete. Syphilis. I'm sharing syphilis with all of you today, okay? So this one is syphilis. If you look really closely, you can actually see here, these little lines, those are the endoflagellum, okay? So this one, you can see those endoflagella, and that's another hallmark of the spirochetes, is that endoflagella that from both of the ends, so those polar ends of the cell, they kind of work their way inward, just underneath a sheath, and their rotation will corkscrew this organism through the media that it's in, okay? All right, any questions on these 4? Let's go ahead 1. Now your turn. Draw 2. So Alcaligenus faecalis and Pyrococcus furiosus, which is one of the coolest, not just because of the name, but because of its nickname. It's the Russian fireball. Okay? Pyrococcus furiosus. Take a couple minutes to draw these 2.
Look at the name.
No. Wait a second. This is bull.
Amphitrachus?
Yeah.
Is there only like one on each side?
That's a great question. Sometimes we'll find just a single one, sometimes you'll find an actual tuft.
Okay.
So in this example here, if you drew one with one on each end or like more than that,
Was there any way to like specify if there's one or more? Or does it
I think you could say like amphitrachus and monotrachus.
You did it wrong.
Amphitrachus and multitrachus.
Okay. Okay.
So the amphitrachus, so there are some that are amphitrachus that aren't spirals, and this is actually an example of one of those. So amphitrachus means flagellum on both ends.
So it doesn't matter the shape?
Nope, nope, it's independent of the shape, it's just referring to the flagellar structure.
Right. You're like asking us to put it in. No, so like for all of like the outline and the bacillus and
Bacilli,
the
yep,
okay.
you're good, you're good.
So like some of them have like multiple like attachment or like the sarcina and stuff. Do you leave out like the main like cocci or bacillus?
So when if you're saying that like what the structure is.
Yeah, so like let's say the instead of coccus, lophotrachus, it would be like staphylo. Would you put coccus in it?
Yeah, you would put, because there are examples in this realm, incredibly rare, of staphylobacilli.
Okay.
So you so the staphylo is kind of like a prefix to add on to bacilli or
So it'd be staphylococcus.
staphylococcus if it were closer.
And then you put lophotrachus, so? Okay.
Yep.
So like all of these are just
Yeah,
like
so like,
prefixes?
yeah, and that's why I put that dash there, to say like, these are ones that you would put above, like, with it to add an additional level of the description.
And then sarcina and tetra, those are just
Okay.
like are those always like circle?
Oh, you're asking if they're always
Here, because these ones,
rods.
because these ones are all
So it's like, I'm definitely not that but like rods, like, you know, during the fishing, rods would also be in the chain, so it would be strapped.
yeah, like dipple shrimps, those are all like sarcina and tetra. So like in the notes, you just have a little dash of that, you know what I'm saying?
Alright. Who wants to draw? Who wants to draw? Yeah? Alright, great. Sure. Alright.
I think my drawing's pretty legit.
Alright, there you go.
Well, I got the magic drawing pad.
Yeah, the magic drawing.
I'm sorry, guys. It's not doing it.
What?
Look, it's like a dot.
There you
Oh,
go, there you go.
There we go.
I was hesitating.
Don't blame the pen.
Takes time.
No. Oh, it's moving.
Well, this is what I spent my summer doing, guys. It's like trying to figure out how to work this, just for a day like today.
Boom. Should I draw more?
Which one is that?
That's the first one, isn't it?
Okay, it's amphitrachus. What's that mean?
So there's a lot.
No. Amphi means what?
One.
Or both, right? Amphi means both. So there's one on each side for this one. If you drew a tuft on each side, that's fine too. I didn't specify whether it's monotrichus or
It looks
lophotrichus.
beautiful.
That is beautiful.
Thank you.
Absolutely. All right, Zach. Russian fireball.
That's a big.
Oops. Yeah, this is a really big one. This can cause serious disease.
Actually, it can't. All right. So pyro means what?
Pyro, fire.
Fire, right? lives in really hot temperatures. That's why it can't cause disease. It doesn't survive at, or it doesn't grow at our body temperature. But it is furious nonetheless, okay? All right. I don't know. It's just the nickname that it got. I don't know exactly. But if any of you would like to see like a cross-stitched one of these, I have one hanging up in my office because that's what my... I know. No. My sister-in-law does, and so she made a whole bunch of these. Okay. There's one of a bacteriophage. There's one of the Russian fireball. Yada yada yada. All right. Right. All right. Let's finish up with a couple of questions here. So we've talked now a bit about this movement and that it requires a flagellum. Typically, the movement of microorganisms is towards a chemical or away from a chemical. If it's moving towards a chemical, typically what that means is that chemical is a chemoattractant. So the microorganism is attracted to it. If it's moving away, chemorepellent. And the decision to move, and I put decision, I'm using this term loosely because it's really not a decision. It's more like an impulse. But that decision to move toward or away is based on receptors that are on the surface of the bacterial cell. So think about if you, for example, are sitting or are cooking something, you grab onto something hot, and immediately your response is to pull your hand away because it starts burning. That's a neurotransmitter response. Bacteria don't have neurotransmitters, but in a very similar way, they do have responses to stimuli, and their stimuli are what's in the environment. So what we will find is that bacteria move in what we call runs and tumbles, towards or away from these various things. Okay, so the movement towards a chemoattractant is going to be based on a couple of features here. So if there is something like serine, an amino acid, or glucose, or something like that, what we'll find is that the microorganism will move towards it in these runs and tumbles. So let's say like that exit sign up there is, I don't know, my glucose. Bacteria are going to move in the following way. They move straight. Okay. Anybody have a Roomba? One of those like...
I can't afford a Roomba.
I can't either. Okay, so if you've ever seen one, they basically move until they run into something. Well, bacterial cells move until the chemoattractant concentration begins to decrease. So if, for example, I'm moving like in this direction here, I would keep going this way until I start to sense that concentration to decrease. The response of the cell then is to undergo a tumble. Essentially, a tumble is this. And it reorients itself. And that movement, that tumble is completely random. Wherever it ends up at the end of that tumble, it starts moving in a new direction. And the cell continues to go through these runs and tumbles until it's reached the highest concentration of the chemoattractant. Over time, runs get shorter, tumbles become more frequent because the changes in concentration gradient as the cell gets closer are more frequent. Chemorepellents, the microorganism moves away from. So as they're sensing lower concentrations, they will continue to go on a run. As soon as that concentration begins to increase again, you'll see a tumble and a movement in a new direction. Obviously, this requires energy, right? Where is it getting its energy for this from? So what do we consider a source of energy in the cell? What was that? ATP? Anybody disagree with ATP? Everybody agrees with ATP. Okay, you're all wrong. In this case, you're all wrong. Zach, you got a different one?
Well, ATP kinetic energy.
Not a bad guess. It's not, though. it's actually, if we think about, go back to in your brain to the time of Gen Bio courses. When we make energy, okay, oxidative phosphorylation and cellular respiration, right? So in that process, you have this ATP synthase. What drives that ATP synthase? Yeah, we'll come back to it. Hydrogen. Okay, hydrogen ions. And it drives a turning movement. It's exactly what happens with the flagella and bacteria. So hydrogen ions will come back in passively through through a little channel that's at the base of the flagellum. That's that. And as those are coming through, this tail or this little piece here spins like a rotor or turbine and creates then the movement for that flagella to whip around. And that's how we get the propulsion. And so the allowance of energy to or the allowance of hydrogen ions to come through that little motor there or to shut it off is dependent again on the chemical signals that the bacteria are sensing in their environment. Okay? All right, one more thing I want to cover today, and that is going back to this structure here. Where does replication and transcription occur in a eukaryotic cell? And this isn't a trick question. It's in the nucleus, right? Where does translation occur? On a ribosome, which is where? Cytoplasm on the ER. I would take cytoplasm as an answer here. Okay? They're physically separated from one another. Anybody disagree with that? Are these physically separated in bacteria? And again, this isn't a trick question. No, they're not physically separated. There's no organelle that's separating the DNA from the ribosome. And so one of the things that's unique in bacteria that is distinct from eukaryotes is in bacteria, we can find transcription and translation occurring at the exact same time. That's not something that can happen in eukaryotes because that mRNA has to be processed. You get rid of the introns. That gets exported out to the cytoplasm where translation occurs. In bacteria, there are no introns. Every single piece of the mRNA that's produced is exon. So what we find in prokaryotes are, at times, folks have looked at these ultra-fine resolution images of transcription and translation in prokaryotic cells, and this is what they observe. This is my DNA here. Here are not ribosomes, RNA polymerase. Here is the mRNA being made. And it creates what they call a Christmas tree light pattern, where you get these mRNA tails of increasing length, and as they're coming off of the DNA, as they're being actively transcribed, you will find the ribosomes starting to associate with them, producing protein or producing an a polypeptide. And so what this allows for in prokaryotes is incredibly quick changes in response to the environment. In a eukaryote, to go from stimulus to changing a gene expression, turning on or turning off a gene and making that protein can take anywhere from 20 minutes to an hour to do before you can detect those changes. In bacteria, 2 to 5 minutes we can detect those changes. Incredibly fast. And this is really important because the environment that bacteria are found in is constantly in flux. Whereas we can regulate our internal environment so the changes are more slow, a bacteria cannot control, for example, this morning I had yogurt, so the lactobacillus in there were refrigerated. They were in a pH 4 solution. And then I ate them, temperature increases dramatically, pH drops from 4 down to 2 in seconds. And so bacteria has to be able to adapt to those changing conditions. And this is one way that allows them to do so. Okay. This is something we'll come back to later on, but I did want to just kind of point this out. Any questions? All right. So for homework, we're going to do another group wiki. So we're going to repeat something that we've done before. There are no pictures associated with this one. So I hope to not get any emails saying, I can't see pictures. What you're going to do, we're going to move into some classical categorization of bacteria, and we're going to focus on what we call the cell envelope. The envelope consists of the cell membrane, the peptidoglycan, and then in some bacteria, an outer membrane and what we call the periplasm. For the homework, you and your group are going to do a wiki that gives some just basic general information on 3 different categories of bacteria. Oh, that's the wrong one. And it's the one labeled as differential staining. So here's the table. What I would like each member of the group to do is to fill in one of these rows for the 3 different types. So we're going to talk about gram-positives, we'll talk about gram-negatives, and then we're going to talk about what we call acid-fast bacteria. And acid-fast include probably the prototypical member of acid-fast bacteria is Mycobacterium tuberculosis, which causes tuberculosis. And so there's some differences in how they stain, the processes by which we stain them, differences in the cell wall, differences in the membrane structure, and whether they sporulate or not. And that's not something we'll cover until Monday, but do take a quick look at that as well. And then we'll go through this on Friday. All right, so that's all I've got. Afternoon, folks. Enjoy lab, and I'll see you all on Friday.